Okay, thank you so much. Our today's talk is about carbonic anhydrase inhibitors and its potential therapeutic applications. Quickly, we will cover certain points regarding the structure and function and localization role of carbonic anhydrase, and also its historical applications as diuretics and its promising recent applications such as cancer and Alzheimer's disease, and also the recent one regarding the global warming, and also some of our contributions in this regard. And finally, but not inshallah, finally, the conclusions and the perspectives which we will try to see.
Dr. Miza, can you view the presentation? Can't see the slides. Okay. Okay, can you see now? Okay.
Yes,
Sorry.
yes,
Very sorry.
we're okay.
Okay, thank you, Doctor. I'm sorry, I don't know if you heard me before or this is the first time to hear.
They could hear, but they couldn't.
They couldn't see. Okay. We will cover carbonic anhydrase regarding the localization, definition, structure, function, and its historical applications as diuretics, and also the recent application as anti-tumor agents, and also for combating global warming, Alzheimer's disease, and other things, in addition to few contributions from our side. And we will see what conclusions and the perspectives could be done. Actually, carbonic anhydrase as an enzyme is a large family of enzymes which are widespread in most of the living cells. And mostly all organisms are containing carbonic anhydrase, and it is non-porphyrin zinc containing enzyme, and it was the first one to be discovered containing metal such as zinc. Does a very simple reaction. It assists the reversible conversion of carbon dioxide and water into carbonic acid, which later on dissociates to hydrogen and bicarbonate. This is very simple reaction, but it is very fundamental in many biological functions inside the cell. And at least there are 5 gene families: alpha, beta, gamma, delta, theta. At least 5. Till now, they discovered the 5. They are unrelated from the evolutionary point of view. Alpha carbonic anhydrase is found in humans, and this is what interested us. Beta carbonic anhydrase and gamma carbonic anhydrase are the main isomers which are interesting these scientific researchers in the field of medicinal chemistry, according to my knowledge. Alpha carbonic anhydrase, which is found in humans, may have about 15 isoforms till now. They discovered 15 isoforms from this enzyme, and it does a very simple reaction, as I said, carbonic hydration of carbon dioxide. How this is done? Actually, the zinc metal in the carbonic anhydrase enzyme plays a very important role in this, because it is linked to 3 histidine residues in the carbonic anhydrase, and the fourth orbital is occupied by water molecule. And we know zinc atom as an atom is electron withdrawing. It withdraws electrons from neighboring molecules and atoms, and so it gets electrons from water and make it easy for water to lose hydrogen and keep OH. Later on, OH adds to carbon dioxide to produce bicarbonate. This is very simple reaction, but it is very important in order to keep the pH buffering system of the cell, and also to make the required ion transport and many other functions we will see just now. So carbonic anhydrase initially was identified in RBCs of cows, but later it was found to be abundant in most of the tissues of mammals, plants, bacteria, and algae, and so on. In order to see how the potential of these 15 isoforms, we will concentrate on 4 of these 15 isoforms, which are interesting according to our research in cancer. Carbonic anhydrase 1, 2, 9, and 12, and also 4 to some extent. In this slide, we will see that the relative activity, affinity, and the kinetics of the catalytic activity of carbonic anhydrase to allow hydration of carbon dioxide varies to varying degrees. Not all the isoforms have the same catalytic activity, and not all of them have the same affinity for the inhibitor. Our inhibitor is sulfonamide, and the affinity and catalytic activity varies a lot. But the 4 which we are interested in could be divided into 2 categories. The first one, carbonic anhydrase 1 and 2, are the housekeeping enzymes. They keep the normal physiological functions of the cell. But carbonic anhydrase 9 and 12 could be considered as the tumor-associated isoforms, which means they are expressed to some extent in solid tumors, and they are not as expressed as they are expressed in the normal tissues, which means they are upregulated in solid tumors, but they are either downregulated or not regulated at all. Carbonic anhydrase 9 is missing in most of the solid tumors, but carbonic anhydrase 12 is not like that. So we can see carbonic anhydrase 2 has high catalytic activity and high affinity for sulfa or for the inhibitor in general. And carbonic anhydrase 9 and 12 are also highly expressed, but we can notice that carbonic anhydrase 8 and carbonic anhydrase 10 and carbonic anhydrase 11, these three are not yet fully understood, and they need to be explored. Okay, carbonic anhydrase 12 has active catalytic site similar to carbonic anhydrase 2, but carbonic anhydrase 2 is considered as a housekeeping enzyme, and 9 and 12 are the tumor-associated isoforms. If we are going to move to the hydration and inhibition via different kinetics, we will find that the prototype sulfonamide compound, which is considered as the standard drug for inhibition of carbonic anhydrase, is acetazolamide. The structure is seen like that. And we will find varying degrees of activity, but as we said, we will be interested in 1, carbonic anhydrase 1, human carbonic anhydrase 1, 2, 9, and 12, and all of them, except 1, is a housekeeping one, is considered to have high activity level and also strongly inhibited by sulfonamide compounds. So we will move to the reactions which are catalyzed by alpha carbonic anhydrase in humans. Hydration of carbon dioxide, hydration of isocyanate, hydration of diamide, hydration of aldehydes, esters, sulfonates, aryl halide or alkyl halides, phenoxyacetyl chloride, and also sulfonyl chloride. And so we can conclude that any compound which contains a similar feature of this or assists reaction on the human cell or in the living cell will be affected by such inhibition. What about the structures of the different carbonic anhydrase? There are claims that they are dimer or they are trimer, but we are interested on carbonic anhydrase 9 and 12, as we said, and both of them are considered to be dimer. in addition to the zinc metal at the middle of the enzyme. And also there are 3 histidine residues linked to the zinc atom, as we said before. And we can notice that carbonic anhydrase 9 is overexpressed in many cancers, but little expressed or not expressed at all in normal tissues. Contrary to carbonic anhydrase 12, which is expressed in normal and tumor cells, but very few solid tumors. It is found on the solid tumor, but not on the normal cell. And this is interesting for the selectivity of the inhibitor, which could be potentiated in this way to eliminate side effects and something like that. So renal cell carcinoma and carcinoma of the cervix, breast, and lung are some of the examples where carbonic anhydrase 9 is not expressed on normal tissues, but elevated on tumor tissues, and so could be considered a suitable target for treating tumors. Also, we have 3 acatalytic isoforms, which is 8, 10, and 11. They are acatalytic. Then we can see now the role of carbonic anhydrase in the living cell. It showed it is present, as we said, in all forms of life in the 3 living domains. Zinc ion complexes the water, as we said before, and so allows hydration by the mechanism illustrated before. And it is very important in the ion transport processes. It maintains the physiological pH and the buffering system. It removes carbon dioxide, and it is very important for the synthesis of the vitreous ocular humor eye secretions. And also it is involved in the ion transport and respiration. This is the first observation to watch with the rest of the researchers. And also it maintains optimum substrates from carbonate and carbon dioxide. In addition to its effect on osteoporosis and the mental retardation, and in the RBCs, in addition to the photosynthesis, ribulose, bisphosphate carboxylase. And also carbonic anhydrase 2 is very essential or very important in Parkinson's disease, Alzheimer's, aging, diabetes, rheumatoid arthritis. All of these areas are yet to be fully understood. What about the historical application of carbonic anhydrase? Historically, it was used as diuretic. Of course, it is not the perfect diuretic, and it needs to be administered with other thiazide diuretics or something like that, because it is found in the nephron, and it by inhibiting carbonic anhydrase, it prevents the reabsorption of bicarbonate. However, reabsorption of sodium occurs far away in the proximal convoluted tubule and something like that. So it is not very effective as diuretic, but it is effective to be used in addition or in combination with other diuretic agents. So acetazolamide is the prototype or the model diuretic drug. And from the pharmacokinetic point of view, we could find that its efficacy and absorbance is good enough, and it affects for about 12 hours in a single dose. And also it reduces the reabsorption by 50%, and so it is a moderate diuretic agent. And also it is very important on the cellular body of the eye, where it reduces the secretion of bicarbonate, and so very important on treatment of glaucoma. In addition, it is used topically, and the topical uses allows avoid the side effects of sulfonamides and the deleterious effects of these compounds. What about the promising clinical application or the recent clinical application of carbonic anhydrase inhibitors, treatment of glaucoma and reducing intraocular pressure? in addition to its use in obesity, where carbonic anhydrase 2 and 5 inhibition allows to lose weight. In addition, in anti-epileptics, carbonic anhydrase 2 and 7 also are important in this respect. Recently, there are some publications regarding the neuropathic pain and sleep apnea, and also Alzheimer's disease and cerebral ischemia, and also immunological function. And the last topic, which is our area of interest, is against cancer by using inhibition of mainly carbonic anhydrase 9, and to some extent carbonic anhydrase 12. And so this is the structure of the enzyme carbonic anhydrase 2, which is important in epilepsy, obesity, and glaucoma. We can find that the 3 histidine residues around the zinc atom in the middle, and they are colored green. These are the 3, histidine 94, 96, and 119. And also there is 64 away, somewhat away, against the zinc atom. And also there is another cluster, histidine 3, 4, 10, 15, 17, which are colored in orange. What about the relation between cancer, carbonic anhydrase, and sulfonamides? We know cancer is a major health problem which concerns the medical community all over the world, and it is supposed or expected and projected to be the leading cause of death, as reported earlier, to be during around 2030, to be a great headache to the scientific community and the medical field. And also cancer has a deleterious effect on the economy and physiology and the lifestyle of the individual and the community as well, because it affects the families and the health systems in every aspect. And currently, the major treatment systems for cancer, radiotherapy, chemotherapy, or genome or something like that, is not highly efficient, and maybe some types of them are highly expensive, which people cannot accommodate. In this regard, we or the scientists offered to use single drugs or multiple drugs, which means to use single drug and benefit from the reduced cost and pharmacokinetic simplicity and enhanced efficacy by using single drug. But using multiple drugs have some side effects, like some drawbacks, such as the complicated side effects, but also it allows targeting many targets, and so could be efficient, but it need to be studied and to see what is the merits of this path or this path. And so the most important point on carbonic anhydrase is the reduced expression on normal tissues regarding carbonic anhydrase 9 itself, and the overexpression on solid tumors. And the beauty of sulfonamide is that it shows, or according to some literature, it is showed to be localized specifically on tumor tissues. Some literatures claimed that sulfonamides specifically concentrate or localize on solid tumors, but not on normal tissues, or not to the same extent of localization. And so if we find some compound, if sulfonamides is able to inhibit specifically carbonic anhydrase 9 or carbonic anhydrase 12 without affecting other isoforms, this will be very good removing or avoiding many serious side effects which could be obtained from such chemical compound. Here we can see the top 5 cancer incidence and mortality all over the world according to the statistics of 2018. As we see, the incidence is 18 million or something like that, The mortality is about 9.6 million, and the prevalence is 43 millions. And the most common cancers incidence is lung, breast, colorectum, and cancer of the colorectum and the prostate. And the mortality also, the highest one is from the lung, colorectum, stomach, liver, and the breast. However, the prevalence is for breast, colorectum, prostate, lung, and thyroid. And so we can see that breast and colorectum could be considered the major regarding incidence, mortality, and prevalence. What about the global new cancer cases incidence? The new global cancer incidence shows that the cancer is increasing, not decreasing. Some types of cancers are increasing, not decreasing, mainly breast and lung, and also colorectum. No, not colorectum, breast and the lung, breast, no, not lung, breast and breast, mainly breast. Okay. What about in Malaysia? The 10 most common cancer incidents in Malaysia is, as you see, breast, colorectum, and the lung also. Breast, colorectum, and lung. What about the incidence, or comparing what is going on from 27 to 2011 compared to 2012 to 2016? I'm sorry, I could be able to get, this is what I can get. It is somewhat not very recent, but it's okay. It shows that if you see to the part of the slide to the right side, where you will find that there is increase in the new cancer cases regarding the breast and also the colorectum, and in the middle it is the lung. It is not the breast. It is the lung, not the breast. All these three, breast, lung, and colorectum, there is increase in the incidence. However, the cervix, the number of cases reduced, and so this is good enough. The cervix is reduced, but the colorectum and breast and lung, all of them show with higher new cases from 27 to 2016. So the Malaysian National Registry report concluded that the percentage of newly detected cancer cases in stages 3 and 4 rose from 58 to mostly 64, comparing between 27 to 2016. And also the new cervical cancer cases dropped. Also, there is a significant increase in breast and colorectal cancer. Okay, so we can see in this slide the distribution of the different isoforms, mainly carbonic anhydrase 9 and 12, comparing normal tissues and the tumor tissues or the solid tumors. The first one is CNS neurons. We could see that carbonic anhydrase 9 is not expressed at all in normal tissues. However, it is highly expressed on the tumor tissues. But carbonic anhydrase 12 is expressed in both, although it is expressed to lower extent on the normal tissues, but it is expressed in both. So it is not good enough to be considered as a target for CNS neurons or glioma. The second one is the esophagus. We can see that carbonic anhydrase 9 is not expressed at all in normal tissues, but it is expressed in solid tumors, extensively expressed. Carbonic anhydrase 12 is not good for this, also for the esophagus. And also the same for the respiratory tract. And we can see on the colon cancer, carbonic anhydrase 9 is expressed to some extent on the normal tissues, but highly expressed on the solid tumor, same like carbonic anhydrase 12. And so it could be considered to some extent suitable for combating colon carcinoma. And the same could be seen for carbonic anhydrase 12. This is the major important point for carbonic anhydrase 12, where it is not expressed at all in the normal gallbladder tissues, but it is highly or somewhat expressed in the biliary carcinoma. Carbonic anhydrase 9 is not useful here. Carbonic anhydrase 9 could be useful also in the pancreas to some extent, but it is very useful regarding the kidney and also the uterine cervix, and it is not expressed in prostate cancer, so even prostate cancer or normal prostate tissues. Endometrium also, it is not expressed at all in normal tissues and expressed to some extent on the endometrium carcinoma. And the breast, it is very interesting. And so we could conclude that breast and CNS neurons, esophagus, respiratory tract, and kidney, and also uterine cervix, carbonic anhydrase 9 is a very good target because it is only expressed on the tumor tissues without the normal ones. Now we have some contribution on this area, and we have synthesized about 16 compounds which were published in the European Journal of Medicinal Chemistry. And we found that 2 compounds were interesting enough to be further studied, which is 3C and 3D. This is the structure of both compounds, 3C and 3D. 3C was found to selectively inhibit carbonic anhydrase 9 and 12 without affecting carbonic anhydrase 1 and 2, which are the housekeeping enzymes. And it also inhibited cancer cell proliferation. By the way, we tested them against colorectal cancer cells. We will see now, we used 2 colorectal cancer cells, and we found that they are able, both of them, or mainly 3C, was able to inhibit the cell viability in a dose and time dependent manner at concentration of 5.45 micromolar, not millimolar, micromolar. I'm sorry, this is not micromolar, it is micromolar. And also it specifically inhibited tumor-associated 9-12 protein expression as well, the protein itself, without affecting the expression of CA1 and 2. And also it induces apoptosis. And we found the mechanism behind this to be increasing ROS levels, B53, and modulating BCL2, BC12, and polyadenosine ribose phosphate and cytochrome C release. And also it reduced NADPH, GSH levels and cell migration. However, 3D was not as strong or as potent as 3C, but it also done the same mechanism because as we see from the structure, the difference is very simple difference between both compounds. This is the results which we obtained at different concentrations of the target compounds. We used all of our compounds, 16 compounds, at the same concentration, 10 micromolar, and we found that 3C, as you see, and 3D are the most interesting of them. And also we tried these compounds. We took 3C itself and tested it at different concentrations, and we found that at 10 micromole, the activity is, it reduces cell viability to less than 20% compared to the standard uncontrolled drug. Cell viability also was found to be affected over time, and we found the maximum inhibition of cell viability happened at after 72 hours. And also it inhibited the cell migration, as we said before, and this is the other proof for what we found. And this was published on the As I said before, the European journal, the first one, the chemistry part was in the European journal, and the biology part was on onco targets, and also 3D was published on onco targets. We found that 3D inhibited cell viability, JAK2, STAT3, and anti-apoptotic proteins. However, it induces ROS and pro-apoptotic proteins and activates caspase cascade. Also 3D, we tested the 3D in combination with doxorubicin against also the same two colorectal cell lines, and it reduces the cell viability. And this work was published on bioorganic and medicinal chemistry. And we could see that doxorubicin alone was not good enough to inhibit the tumor cells, or its effect is activated or potentiated, or there is a synergistic effect when we combine 3D with doxorubicin. So doxorubicin could be suitable for this work. Also, this is the crown of our work. We were able to synthesize 3 groups of compounds. The first one, we can consider it phenyl ethyl amine derivative, phenethylamine. But this, we will find later the efficacy of this compound compared to the other 2 sets. The other set is the meta-carbonic anhydrase quinazoline derivative, and the third one was the para one. So we have 3 keys. We need to focus on these compounds for some extent. The first one, this compound and these derivatives, also about 16 compounds, very simple compounds, could be obtained by one-step chemical reaction, and they contain the thiol moiety or the thiohydryl moiety, which is very important on inhibition activity of carbonic anhydrase and also the sulfonamide group. We found this series of compounds, but actually they are not interesting. Why? Because if we look at the carbonic anhydrase 12 inhibition and compared it with the inhibition of CA2, the housekeeping enzyme, we will find that this group of compounds inhibited both to mostly similar extent, and so it will induce several side effects. It is not specific for carbonic anhydrase 12, and unfortunately at that time we were unable to find or to buy carbonic anhydrase 9, so it was not tested for these compounds. Maybe we will find some importance if we found this carbonic anhydrase 9. So let's move to this second series, which is the para compounds. We will find that we obtained interesting compounds, but also the selectivity for CA9 or CA12 is not enough compared to the activity against CA2. So also this group of compounds is not fortunate enough, or we were not fortunate enough to get good compounds. Although there are some examples which show high or activity at low concentrations, but they were not good enough for us to proceed for further studies. But this one is the crown of all of these compounds, which is a meta-sulfonamide linked to the quinazoline with the SH group. We will see the interesting thing here. If we look at compound AA67, we will find that it inhibited carbonic anhydrase 2 at concentration 370. However, it inhibited CA12 at 0.48. And so it is highly selective for carbonic anhydrase 12, and it is not suitable to inhibit carbonic anhydrase 2. And the selectivity ratio here is 770. So it is highly specific compared in comparison between CA2 and CA12. Also, AA70 compound AA70. For inhibition of CA2, we need 175 compared to 0.58 concentration to inhibit carbonic anhydrase 12. And the selectivity ratio here also is about 300. Regarding AA73 and AA74, both of them have selectivity ratio around the same like AA70. And so we can consider these 4 compounds to be very interesting compounds, although we didn't test these compounds against CA9, but the results in our hands now are good enough to consider these compounds very suitable for structural modification and studying what is going on and seeing what could be benefited, or how can we benefit from these compounds. And so we moved to the molecular modeling studies to see these 2 compounds. And by the way, from the chemistry point of view, we may expect to find all of these compounds in one of 2 forms, imino form or thione form, where the hydrogen is fluctuating, moving between the nitrogen and sulfur and so on. And also the same for the meta compound, which is the most interesting one, and for also the phenethylamine derivative. And so we tried to see virtually what will be the situation with carbonic anhydrase. And so we may expect 2 conformations. These 2 conformations, the first one where the NH2 is oriented in the same side with SH. And so the distance between both moieties will be somewhat smaller than the distance if the NH2 took the opposite side from the SH. In this case, we will have longer distance between the SH and the NH2, and also the rest are the same, but these are the main possible conformations for these 2 compounds. And so we used acetazolamide superimposed with our compounds regarding the crystal structure of CA9 to see what is going on. We found that the 2 compounds aligned by superposing the SO2 NH2 and the aromatic rings. So we will find the quinazoline moiety is protruding outside. Oh, sorry. So we can see that the carbonyl of quinazolone may establish bridged hydrogen bonding in presence of water. In presence of water, the carbonyl group of quinazolone may establish hydrogen bonding in presence of water with GLN67, and the distance, as we see, 4.6 or something, around 5. And also we can see that the quinazoline aromatic part is oriented toward the solvent and could be considered hydrophobic. And also there is a small hydrophobic portion, which is the SH group protruding towards leucine. And so we can expect these compounds to be very, very interesting on modification in order to move forward to see what they will do with solid tumors, specifically breast, lung, and colorectal, which are highly expressed, and there are new cases specifically in Malaysia. The last part of the application of carbonic anhydrase is combating global warming. And so they are suitable for reducing carbon dioxide emission from the industry. In this regard, we have two strategies could be used. Either to use the official or the old-fashioned mode of using the sequestration of carbon dioxide by usual means, by chemical means, which is very, very tedious, expensive, highly expensive, and very costly. And so it is not suitable enough. It is not suitable enough to capture carbon dioxide by chemical means. All of you know, of course, the problem of global warming and its effect on the different aspects of our globe. And so,
Sorry, Prof.
Sorry.
We cannot see anything on the slide, on the
Now?
screen. Yeah.
I'll help you out. Just give me a second, please.
They told me that Chrome is the best.
No problem. No problem. Were you here? Can you view the PowerPoint now? Presentation?
Yeah, we can see it.
All right. Okay.
Thank you.
Sorry.
No, I'm sorry. I don't know if this one slide, two slides, or more are missing. Dr. Miza?
I think they're okay now.
Okay, so mitigating or reducing global warming. Good. Okay, thank you.
Can we put it a presenter view, please, Prof?
It is on presenter view right now.
From our side is the slide view.
Okay. Is it okay now?
No, not really, but maybe we can just enlarge the slide so that we still can see it.
Okay.
Yeah.
Can you see? All right. Okay.
We can go then.
Yeah, I have to present like this. Yeah.
Okay, okay. Jazakallah.
All right.
Okay, so mitigating global warming could be done through one of two strategies. The first one is the sequestration or storage of carbon dioxide or carbon atom itself. But this way proceed through a very costly pathway, and the byproducts are not as interesting as the other one. So this is grave demerits of carbon capture sequestering technology. It is highly expensive. It takes about 25% from the power of the plant, and also the solvent used is highly corrosive, and it is very difficult to keep all of these carbon dioxide and pipes underground. So the other way is the most interesting one, is to use carbon dioxide, to use carbonic anhydrase itself in order to sequester carbon atom itself and the carbon dioxide, similar to what is going on photosynthesis. By this way, we will get many advantages. The first one of them is this procedure is eco-friendly and adaptable, and also the after sequestering carbon dioxide by carbonic anhydrase, we will get very useful byproducts such as calcium supplements, mineral carbonates, and also antacids, which are useful, and also excipients for pharmaceuticals, and also as remediation for feed stocks and construction materials such as gypsum and cement and something and many other things which could be so useful. By this way, we could add carbonic anhydrase in different places, and it will by certain means, by using magnesium and calcium oxides, zinc also as well. In this way, carbon dioxide could be converted to carbonates, and so instead of regular industrial, expensive, and inefficient customary ways, we could obtain the sequestration of carbon by this means. Finally, we can conclude that we need to modulate sulfonamides in order to be specific for certain isoforms of the enzyme, and also new lead compounds are in need to be obtained by combining these inhibitors which we obtained in the final slides to be joined to some natural products or some secondary metabolites or something like that. And also further experiments in vitro are needed, and also extra running or extra tuning for the specificity of these carbonic anhydrase, and also in addition to find the suitable biotechnologic application in order to get some compounds which could be, or some strategies for application on global warming, for example, or against cancer or something like that. Finally, I want to say I'm very sorry for what happened, but this was not expected, and I'm very thankful to the organizing committee, to our colleagues, and a special thank for Dr. Miza, Dr. Gourmet, which helped me a lot today, and also Dr. Fazlin. Thank you very much.
Thank you so much, Prof. Alafeefy. We are open for question and answers. Anyone wants to ask question to Prof. A? We at the Faculty of Pharmacy call him Prof. A. Right, Prof? Any question?
Okay, Assalamualaikum warahmatullahi wabarakatuh.
Alaikum assalam wa rahmatullahi wa
Prof, I'm Amin, lecturer from Faculty of Pharmacy, UiTM, from campus Bertam in Penang.
Welcome.
Okay, I would like to ask a question. Before that, I would like to congratulate you for the very interesting presentations. It's indeed a lot of works you have done.
Thank you so much.
Okay, I just curious one thing, how the sulfonamide moieties could affect, and how could affect the carbonic anhydrase. I mean, how the sulfonide group interact with the carbonic anhydrase specifically. That's the question.
Thank you very much. Very interesting question. And I could say, or not could say, this is what reported in the literature, that as we said before, zinc enzyme contains vacant orbital. And when it interacts with carbonic anhydrase, 3 histidine residues are interacting with the zinc metal, and the 4th orbital is occupied by water. And so carbonic anhydrase inhibitors act by one of 2 mechanisms, either addition or substitution, which means they either add to the carbonic anhydrase zinc with water, and so they complex water from leaving or from liberating the hydrogen and adding OH to carbon dioxide to produce bicarb and hydrogen, or either by substituting water. And so it removes the water which carbonic anhydrase uses to hydrate carbon dioxide. I wish I explained your question.
Thank you, Prof. Yeah,
Thank
as I know, the carbonic anhydrase have many active sites and loop sites. So I thought maybe it sulfonamides well well suited to the loop site on the active on the carbonic anhydrase.
Well,
Is it?
again, please.
I mean, carbonic anhydrase have many active sites
Yes,
on the structures, right? So is
yes,
that possibilities of the sulfonamide functional group react at the active sites of the carbonic anhydrase?
yes. Mainly, the carbonic anhydrase inhibitor targets zinc itself.
Oh, I
Because,
see.
yeah, because you know, SO2, two oxygens, NH2, also all of them are full of electrons, and zinc metal is electron deficient. That's why they target zinc, and in addition, the water, which helps, which is helped by zinc itself to add to carbon dioxide. So you are correct. It could target different sites, but the most active one to be interacted with is zinc itself and water.
I see. All right. Thank you, Prof.
Thank you so much.
Prof, a question from me.
Okay.
How far do you see that? I see the potential of these CAI to be in for chemotherapy, right? Do you see how far can you expect it to be into clinical trials?
This this needs more efforts. Yes, you are right, but it needs more effort in order to study the other side effects, because all of the work which we published till now, we didn't go for safety studies, toxicity studies, and so on. So the first step, I suggest the first step is to go for the safety for these compounds, but I suggest, in my opinion, it is better to take the meta derivatives which we showed in the last slides, which are for which we didn't carry out carbonic anhydrase 9, and also we didn't go for any tumor cell line. After this effort, we may go for other further studies, inshallah.
Okay, is there any question? Dr. Sadia would like to ask a question to Prof. Alafeefy.
Please. I didn't hear.
Prof, while waiting for someone else to ask question, do you see how big an impact is this study going to be for the therapy?
The impact in the therapy is, we can say that few cell lines could be studied. As we said, breast, cervix, colorectal cancers are the most important area to be for our compounds to be applied, because as we saw in the PPTs, that these cell lines are free, devoid of any carbonic anhydrase 9 on normal tissues, and they are overexpressed on the solid tumors. So if we found the suitable compound which selectively targets CA9 without other side effects, it will be very, very good.
Okay, is there any question?
No.
Okay, Prof, I think, Amin?
Prof, just another one question that I
Okay.
concern regarding your complex on AA27, if I'm mistaken. The compound that you have include fluorine as the R group.
Okay, just a minute.
That you show the structure.
Yes. Yeah.
Right. So I have a concern that the compound might have high level of toxicity. Instead,
This A66, you mean?
yeah, the compound that you substitute the R with fluorine.
Yes.
Yes. So how about the toxicity of the compound?
First of all, this compound is not interesting. Why? I can share the slides with you. You can notice that the activity against CA2, the concentration is 9.4 micromole. And if we try to see the selectivity ratio for this compound, we will not find it interesting. We are searching for, oh, sorry, yes, you are right, for fluoro, 67.
Fluoro. Yeah.
Yes, you are right. We need to study the toxicity here. Yes, you are right. The selectivity ratio is high, but we need to see the selectivity ratio, and also the improvement of this compound could be through changing the fluoro to other atoms which may not affect. Or at first we need to study what's going on.
All right.
But you know, fluoro is present in many interesting compounds such as fluorouracil, and also sodium fluoride for treatment of epilepsy, and many other. But you are correct. Fluoro or halogen in general are not suitable for the ozone layer and the other side effects, and better to be avoided. Yes, you are right.
All right. Thank you, Prof.
Okay, thank you so much, Prof, for the talk. It was a very interesting one. Okay, thank you everyone for joining us. Hope we can meet again in another series of webinar from Faculty of Pharmacy. I would like to thank also the Dean, Professor Madia, Dr. Shariza Saudin, Dr. Gurmit, Dr. Fazlin, and everyone involved, and mostly Prof. Alafeefy. Thank you so much, everyone. Thank you. See you.
Thank you so much.
Bye.